Welcome back. Today we will discuss one important topic in molecular biology, that is DNA replication. In last lecture we have discussed about the experiments that proved that DNA replication is semi-conservative in nature. So today we will discuss the mechanism of DNA replication. Let's start. Firstly, what are the basic requirements for DNA replication? So it requires firstly the substrate, which are the 4-deoxyribose nucleotide triphosphates. Commonly which are present in DNA are deoxyribose adenosine triphosphate, then deoxyribose guanosine triphosphate, deoxyribose cytosine triphosphate, and deoxyribose thymidine triphosphate. So these 4 basic deoxyribonucleotides are involved in the synthesis of DNA. Next comes template. So the parental strands or DNA, both strands of DNA acts as templates for DNA replication. Enzymes and proteins which are involved in DNA replication. Firstly, DNA B protein, it acts as a helicase. It has DNA unbinding property. It helps in uncoiling or unbinding of DNA double helix. Then comes RNA primase, which helps in formation of RNA primer or adds short sequences of ribonucleotides. Then topoisomerase 1 and 2, which is also known as gyrase, it helps in relaxing the tension which is developed behind the replication fork. और ये निक बना के means it can break the phosphoester bonds and can relax the force or tension which is generated behind the replication fork. Then comes DNA polymerases. So there are different types of DNA polymerases which helps in DNA replication and proofreading. Finally comes DNA ligase, which is known as molecular glue. It helps in joining the 2 DNA small segments which are known as Okazaki fragments. So जो Okazaki fragments हैं उनकी joining में enzyme help करता है. Next comes DNA A proteins, then SSB proteins which are known as single strand binding proteins. ये जो proteins होती हैं, ये जो 2 strands हैं unwinding के बाद बने हैं, उन्हें stability provide करता है. Then comes terp binding protein which helps in termination of DNA replication. So these are the basic requirements of DNA replication. This slide shows enzymes and proteins which are involved in DNA replication. Firstly, we have DNA B protein. It is also known as helicase. It helps in unwinding of DNA double helix. Next, primase, which is also known as DNA-dependent RNA polymerase. It helps in synthesis of RNA primer, which is a short segment of RNA or piece of RNA, which is required for the DNA replication. Then comes DNA topoisomerase 1 and 2. This enzyme basically helps in removal of or relaxation of torsional strain which is developed behind the replication fork due to unbinding of DNA. So it relieves torsional strain by cutting and joining single strand, that is topoisomerase 1, or both the strand by topoisomerase 2, which is also known as DNA gyrase. Next, DNA polymerase, which helps in DNA chain elongation. Basically polymerization में help करते हैं. ये DNA का जो नया strand बनता है उसकी polymerization में help करते हैं. And it also has DNA repair activity and proofreading activity. Then DNA ligase, which is also known as molecular glue, which is known to join the Okazaki fragments. जो Okazaki fragments हैं जो lagging strand में बनते हैं उनको join करने में help करता है. Then DNA A protein, it again helps in opening of this DNA double helix. जो open duplex है उसको open करने में help करता है. Then SSPs, which are known as single strand binding proteins. ये बाइंड करती हैं सेपरेटेड सिंगल DNA strands को और स्टेबलाइज करती है इसको. देन टर्ब बाइंडिंग प्रोटीन्स, इट प्रिवेंट हेलिकेज फ्रॉम फर्दर अनबाइंडिंग. जो हेलिकेज एंजाइम है उसको फर्दर अनबाइंडिंग से ये रोकता है एंड फैसिलिटेट्स द टर्मिनेशन, और टर्मिनेशन को ये फैसिलिटेट करता है, हेल्प करता है. This table shows the differences between DNA polymerases of prokaryotes and eukaryotes and their major functions. In case of prokaryotes, there are only 3 types of DNA polymerases, that is DNA polymerase 1, 2, and 3 having different function. Firstly, DNA polymerase 1. In case of prokaryotes, it helps in gap filling and synthesis between Okazaki fragments of lagging strand, while this function is performed by DNA polymerase alpha in case of eukaryotes, and it has primase activity. Then next, in case of eukaryotes, there are some other enzymes, that is RNase H and FEN, that stands for flap endonucleases. As we know that DNA polymerase alpha, it lacks 5' to 3' endonuclease activity. So eukaryotes needs RNase H enzyme for removal of RNA primer from DNA-RNA hybrid. And this FEN1, that is flap endonuclease 1, it is responsible for cleavage of terminal bond between RNA and DNA. So जो terminal bond present होता है RNA और DNA के बीच में, वो remove करता है FEN1, that is flap endonuclease. Next comes DNA polymerase 2. In case of prokaryotes, this enzyme helps in DNA proofreading and repair. So ये इसके दो important function है, and this function in case of eukaryotes is performed by DNA polymerase epsilon. In addition to this, there is another DNA polymerase, that is DNA polymerase beta in case of eukaryote, that helps in DNA repair. And next type of DNA that in case of eukaryote, that is DNA polymerase gamma, which is present in the extranuclear environment, that is in the cytoplasm of the cell, and it helps in mitochondrial DNA replication. So this is very important function which is played by this DNA polymerase gamma, which helps in replication of mitochondrial DNA. Then comes DNA polymerase 3. In case of prokaryotes, this enzyme helps in polymerization of leading strand and lagging strand. So replication of leading and lagging strand is performed by this enzyme, that is DNA polymerase 3. In case of eukaryotes, this function is performed by DNA polymerase delta. So these are summarized functions of respective DNA polymerases in case of prokaryotes and eukaryotes. Next comes RNA primer. It is a short segment of RNA that consists of 5 to 50 nucleotides in length required for synthesis of our DNA. It is synthesized by DNA-dependent RNA polymerase called primase, which has 5' to 3' direction activity using DNA as template. So this small piece of RNA is required for DNA replication. This small piece of RNA provides free 3-OH group for DNA polymerase. So enzyme primase in association with SSB, that is single cell binding protein, single strand binding proteins, form a complex called as primosome and produces RNA primer. So this primase helps in formation of this short segment of RNA primer, and it provides free 3-OH group for the activity of DNA polymerase 3 in case of prokaryotes. Next, steps of DNA replication. So there are basically 3 steps which are involved in DNA replication. First is initiation. It occurs at the origin of replication. So there is a specific site on DNA from where the DNA replication starts, which is known as ORI, origin of replication. For this, it involves unwinding of double-stranded DNA. So जो double-stranded DNA है उसकी unwinding होती है and synthesis of primer, that is our piece of RNA or segment of RNA. Then comes primosome. It is a complex formed by protein and act as an initiation factor. So ये जो primosome बनता है, it starts the initiation of DNA replication. Second step is elongation. It involves addition of new nucleotides, that is deoxyribose nucleotides, based on complementarity of template strand. So जो DNA का parental strand है उसके basis पे नए nucleotides add किए जाते हैं complementary, which forms phosphodiester bonds, correct the mismatch basis extending the DNA strand. Phosphodiester bond add किए जाते हैं between 2 nucleotides. Then there is the formation of replisome. It is a protein complex associated with a particular DNA structure to unwind DNA and synthesis of daughter strand. Then the third step is termination, which means the stop of the DNA replication occurs at a specific termination site. And for this, special protein is required, जिसे कहा जाता है TUS protein, and there is TER site, that is termination site. So these 2 factors, mainly the TUS protein or termination site, these 2 factors plays an important role in termination of DNA replication. So next comes mechanism of replication. Firstly, in case of prokaryotes and in eukaryotes, the replication of DNA is semi-conservative type. It means the one strand is of parent strand and one strand is synthesized new, that is daughter strand. So replication occurs in a series of segments called replicons. Replicons कहा जाता है जिसमें replication होती है ऐसे segments में. It starts at origin of replication. ORI में start होता है. For this initiation, proteins identify specific base sequences on DNA, called sites of origin. तो इनिशिएशन जो प्रोटीन्स हैं वो डीएनए के ऊपर स्पेसिफिक बेस सीक्वेंसेस को आइडेंटिफाई करती है जहाँ से डीएनए रेप्लिकेशन शुरू होता है, दैट इज कॉल्ड साइट ऑफ ओरिजिन. एंड इन केस ऑफ प्रोकैरियोट्स देयर इज सिंगल ओरिजिन साइट. प्रोकैरियोट्स के केस में एक ही ओरिजिन की साइट होती है एंड हैविंग 245 bases which are 80 rich sequences, that is adenine or thymine rich sequences. For example, in case of E.coli-oriC, origin of replication C. While in case of eukaryotes, there are multiple sites of origin. Eukaryotes के केस में बहुत सारे sites होती हैं origin के. For example, in case of yeast, there is ARS, that is autonomously replicating sequences. So these are some features of starting at origin of replication in prokaryotes as well as eukaryotes. First step in case of prokaryotic replication is initiation. This initiation starts from a point which is known as origin of replication, which is recognized by certain proteins जिन्हें कहा जाता है DNA big proteins. These proteins have helicase activity, DNA helicase की तरह काम करती हैं ये, और these helps in unwinding of DNA strands. So ये जो DNA helicase है ये डबल स्ट्रैंडेड DNA को unwind करता है, open करता है from the origin of replication. For this process ATP is utilized. ATP energy चाहिए. This process is energy dependent. As the DNA opens up, जैसे DNA open होता है, Y-shaped structures called replication forks are formed. So Y-shaped structure बनते हैं जिन्हें कहा जाता है replication forks. तो दो बनते हैं which moves in opposite direction. So this structure is known as replication fork, जो Y-shaped structure है. And two replication forks are formed at origin of replication. दो replication forks बनते हैं and these gets extended bidirectionally as replication proceed. और ये दोनों ही bidirectionally move करते हैं, proceed करते हैं in opposite direction as replication proceeds. Single strand binding proteins maintain single strands. इसके ऊपर लगती हैं proteins जिन्हें कहा जाता है single strand binding protein, जो single strand की identity को maintain करती हैं. They prevent from the strand to mix again to form helical structure. तो ये single strand binding protein maintain करती है single strands को. Then there is another enzyme that is RNA polymerase that helps in synthesis of short segment of RNA, that is RNA Primer. Next, DNA polymerase is able to add nucleotides only in 5' to 3' direction. जो DNA polymerase है वो सिर्फ 5' to 3' direction में ही nucleotides को add कर सकता है in the new DNA. It also requires free 3' OH group. This is the very important requirement of this DNA polymerase 3. So for this, the free 3 OH group which is provided by the RNA primer to which it can add nucleotide is free, and में ये nucleotides add कर सकता है DNA polymerase by forming a phosphodiester bond between the 3 OH group and 5' phosphate group of the next nucleotide. So जो दो nucleotides हैं उनके बीच में जो bond होता है that is known as phosphodiester bond. So it occurs between the 3 hydroxyl group of a sugar and a phosphate group which is at the 5th position of the next sugar. So दो जो nucleotides हैं that is sugar, phosphate and nitrogenous base. So these two base nucleotides are joined together by this phosphodiester bond. This essentially means that it cannot add nucleotides if a free 3 OH group is not available. So that's why RNA primer is needed for the DNA replication. Anti-parallel strands replicated simultaneously. दोनों ही strand में replication simultaneously होती है. एक में continuous होती है जिसे कहा जाता है leading strand. Here the synthesis is continuous, that is 5' to 3' direction, and in lagging strand synthesis is in fragments, that is again in 5' to 3' direction में बनते हैं, but जो DNA के pieces बनते हैं they are in fragments और उन्हें कहा जाता है ओकाज़. Next, DNA replication. DNA replication is semi-discontinuous type. It means in one site, that is 5' to 3' side, it is formed continuous, and this strand is known as leading strand, while in the other direction where the parental strand is from 5' to 3' direction, it is formed in opposite direction, that is 5' to 3' direction, and it is found in fragments. So that's why this DNA replication is known as semi-discontinuous. It means it is in one strand it is continuous and in another strand it is discontinuous. So that's why DNA replication is semi-discontinuous and semi-conservative in nature. Here one parental strand is retained and another strand is formed brand new, that is daughter strand. Here DNA polymerase 3 synthesizes most of the DNA, so it helps in polymerization. Then DNA polymerase 1, which has proofreading activity, it fills the gap between Okazaki fragments. And DNA ligase, which is also known as molecular glue, it seals the nick between fragments. So fragments जो DNA fragments हैं उनको जोड़ने का काम करता है DNA ligase. RNA primer and its function that we have already discussed. RNA primer is a short segment of RNA that consists of short RNA sequences that provide a starting point for DNA synthesis. Means it provides a free 3OH group for DNA polymerase. A primer must be synthesized by an enzyme called primase, which is a type of RNA polymerase before DNA replication can occur, that we have already discussed. Important enzymes of DNA replication, again, first is topoisomerase. It prevents the torsion which is developed behind this replication fork. So यहाँ पे जो topoisomerase enzyme है ये relieve करता है tension को जो develop होती है यहाँ पे. Helicase, it helps in separation of DNA strands to those strands हैं. This is DNA B protein which acts as helicase. It helps in uncoiling of unwinding of DNA double helix. And primase, it helps in formation of RNA primer. Single strand binding proteins that prevent re-annealing of single strands. जो single strands हैं उनकी आइडेंटिटी को ये maintain करते हैं, stability provide करते हैं ताकि फिर से re-anneal ना हो. DNA polymerase 1 and 3 synthesizes new strand, and DNA ligase seals the nick via phosphodiester bond. So it is a molecular glue that helps in joining these fragments. Second step in case of DNA replication is elongation. Here DNA polymerase attaches itself to the unzipped two strands of DNA which act as template strands, जिन्हें template strand कहा जाता है और parent strand भी कहते हैं, and start synthesizing new DNA, और नया DNA बनाने start करते हैं in opposite direction. One is leading strand, other is lagging strand. DNA polymerase is only able to extend the primer by adding free nucleotides to 3' end. So जो RNA primer है वो provide करता है 3OH group जहाँ पे DNA polymerase ऐड करता है नए डिऑक्सी राइबो न्यूक्लियोटाइड्स, जो न्यूक्लियोटाइड्स हैं उनको ऐड करता है. वन ऑफ द टेम्पलेट्स रीड्स इन अ 3' to 5' डायरेक्शन. जो एक पेरेंटल स्ट्रैंड है वो रीड करते हैं 3' to 5' डायरेक्शन और दूसरा रीड करते हैं 5' to 3' डायरेक्शन. दिस न्यूली फॉर्म्ड स्ट्रैंड इज रेफर्ड टू एज लीडिंग स्ट्रैंड. जो 5' to 3' डायरेक्शन में नया स्ट्रैंड बन रहा है, दैट इज दिस वन 5' to 3' डायरेक्शन. दिस इज फॉर्म्ड कॉन्टिन्यूसली और इसे कहा जाता है लीडिंग स्ट्रैंड. Along this strand, primase only needs to synthesize an RNA primer once at the beginning to initiate DNA polymerase activity. So इस strand पे जिसमें leading strand बनता है उसमें सिर्फ एक बार ही RNA primer की ज़रूरत पड़ती है, and this is found by this primase enzyme only once at the beginning to initiate the polymerization process by DNA polymerase, जो कि deoxyribonucleotides ऐड करता है complementary to the template strand. The other template strand, that is the lagging strand, is anti-parallel to the first strand and is therefore read in a 5' to 3' direction. ये भी 5' to 3' direction में read किया जाता है. Here RNA primers are added to newly exposed bases on the lagging strand and DNA synthesis occur in fragments. इस strand पे जो DNA है वो fragments में बनता है and but जो direction होती है that is 5' to 3' direction. और ये जो फ्रेगमेंट्स बनते हैं उन्हें कहा जाता है ओकाजाकी फ्रेगमेंट्स, एंड इट रिक्वायर ईच सेगमेंट ऑफ डीएनए सिंथेसिस रिक्वायर फर्स्टली अ स्मॉल पीस ऑफ आरएनए दैट इज आरएनए प्राइमर. हर एक डीएनए के पीस बनने से पहले एक आरएनए प्राइमर की जरूरत पड़ती है, एंड दिस प्रोसेस इज आल्सो इनिशिएटेड बाय दिस आरएनए पॉलीमरेज. सो आरएनए पॉलीमरेज synthesizes the new DNA in both the strands simultaneously, and these small fragments which are formed on the lagging strand are known as Okazaki fragments. इन्हें Okazaki fragment कहा जाता है. And the last step in case of DNA replication is termination. In prokaryotes, for example, in case of E. coli, termination is signaled by specific sequences, जिन्हें कहा जाता है TER sequences, which serves as a binding site for some special proteins which are known as TUS proteins. So TER sequences और TUS protein ये दोनों एक complex बनाती है that leads to termination of DNA replication. So this TUS protein binds to TER sequences to form Tuster complex and stops the DNA from unwinding, जिससे DNA की further unwinding नहीं हो पाती है DNA जो replication fork है वहाँ से आगे. So this is a TUS protein and these are TER sequences. ये दोनों combine करते to form a complex. This stops the movement of replication fork and stops the process of DNA replication. This is termination in case of DNA replication. Next comes end replication or telomere replication. So जो telomeric region होता है chromosome का, that is tips, वहाँ पर replication थोड़ी सी different होती है normal replication से. Replication of telomere is independent of normal DNA replication. Independent होती है normal DNA replication से. So this region of chromosome, that is the tips, इसे telomere कहते हैं. और यहाँ पे जो DNA replication होता है, that is slightly different from the normal DNA replication. Telomere contain RNA proteins. जो टीलोमेयर रीजन है इसमें आरएनए और प्रोटीन प्रेजेंट होता है. सो इट इज़ बेसिकली नॉन एज राइबोन्यूक्लियोप्रोटीन. और यहाँ पे काम करता है एक आरएनए डिपेंडेंट डीएनए पॉलीमरेज. सो दिस रेप्लिकेशन इज कैरिड आउट हियर बाय आरएनए डिपेंडेंट डीएनए पॉलीमरेज बाय यूजिंग आरएनए एज टेम्पलेट. सो जो आरएनए यहाँ पे होता है उसको एज अ टेम्पलेट यूज करते हैं ये एंजाइम विच इज़ आरएनए डिपेंडेंट एंड elongates the DNA by using RNA as template strand. It recognizes G-rich region, that is guanine-rich region, and the tip. So जो guanine-rich region होता है यहाँ पे tips में, so it is recognized by this enzyme and it elongates the strand in 5' to 3' direction. और इसके अलावा यहाँ पे present होता है एक 3' OH group at the terminal of the GT-rich hairpin as primer. ये चीज़ यहाँ पे होती है and extend the existing strand. Here, after subjecting the DNA strand for several rounds of extension by telomerase, जो telomerase enzyme है वो इसमें बहुत सारे rounds लेता है DNA की extension की, and the replication of lagging strand at the end of chromosome is said to be completed. और जो lagging strand है उसकी replication भी complete हो जाती है finally by this telomerase enzyme. So this enzyme helps in replication in case of telomere, that is the ends of chromosomes. So it is different from that of the normal replication. Next step is removal of RNA primer from the newly synthesized DNA in case of leading strand as well as lagging strand. So in lagging strand, two enzymes are needed to achieve this. One is RNase H that removes RNA primer at the beginning of each opposite fragment, and then next comes DNA ligase जो ज्वाइन करता है रिमेनिंग फ्रेगमेंट्स को डीएनए के टू फॉर्म अ कंप्लीट स्ट्रैंड. सो दीज टू एंजाइम्स आर रिक्वायर्ड फॉर दिस प्रोसेस. नेक्स्ट कम्स प्रूफ रीडिंग एक्टिविटी बाय डीएनए पॉलीमरेज 1. डीएनए रेप्लिकेशन इज हाईली एक्यूरेट प्रोसेस बट समटाइम देयर मे बी सम मिस्टेक्स बाय डीएनए पॉलीमरेज बाय इंसर्टिंग सम रॉन्ग बेसिस. कई बार जो डीएनए पॉलीमरेज है कुछ एक रॉन्ग बेसिस ऐड कर देता है कॉम्प्लीमेंट्री to the parental strand के जो sequences हैं उनके complementary sequences ऐड कर देता है. For example, in this case, this parental strand has guanine और जो नया strand बनता है उसमें जो DNA polymerase है वो thymine ऐड कर देता है. So this leads to mismatched base that can leads to serious damage or disease. Most of the mistakes are corrected by DNA polymerase 1. जो DNA polymerase 1 है वो correct करता है mismatch repair को by proofreading the base that has been just added. In proofreading, the DNA polymerase 1 reads the newly added base before adding the next one. जो DNA polymerase 1 है वो चेक करता है जो previously add किया है base वो correct है या नहीं है, and after checking this the new base is added. So a correction can be made अगर कोई गलत base add हुआ है तो. If it is the right base then the next nucleotide is added. So DNA polymerase has proofreading activity. Here the 3' to 5' exonuclease activity of DNA polymerase 1 removes nucleotide from 3' end of the growing DNA chain. So जो DNA polymerase 1 है इसकी 3' to 5' exonuclease activity होती है and और ये चेक करता है 3' end पे कोई गलत base तो ऐड नहीं हुआ है, अगर हुआ है तो उसको ये रिमूव करता है. Next, if an incorrect base has been added, the enzyme makes a cut at the phosphodiester bond and releases the wrong nucleotide. This is performed by exonuclease action, और ये perform करता है exonuclease action of DNA polymerase 3. Once the incorrect nucleotide has been removed, a new will be added again. In E. coli, after replication, the mitogenous base adenine acquires a methyl group. The parental DNA strand will have methyl group, whereas the newly synthesized strand lack them. जो पेरेंटल स्ट्रैंड होता है डीएनए का उसमें मिथाइल ग्रुप प्रेजेंट होता है, वाइल जो डॉटर स्ट्रैंड बनता है नया उसमें मिथाइल ग्रुप एब्सेंट होता है. सो दस जो डीएनए पॉलीमरेज है ये आइडेंटिफाई करता है जो नया सिंथेसाइज्ड स्ट्रैंड है जिसमें मिथाइल ग्रुप एब्सेंट होता है, वाइल इज दिस पेरेंटल स्ट्रैंड कंटेन मिथाइल ग्रुप जिसमें एडेनिन जो होता है वो मिथाइलेटेड होता है. And after this, जो DNA polymerase it is able to remove the wrongly incorporated bases from the newly synthesized strand, which are non-methylated. So it acts as a identifying marker for this enzyme DNA polymerase, जिससे ये नए जो strand है synthesize हुआ है उसको identify करता है और उसमें जो incorrect bases है उनको remove करता है. After this proofreading, जो daughter strand है वो उसमें भी methylation होती है adenine में. So by this process, proofreading is done by these DNA polymerases, that is DNA polymerase 1 and 3 respectively. Next comes the concept of central dogma. This concept was proposed by Francis Crick in 1958. What is this concept? It is the unidirectional flow of information from DNA to RNA and RNA to protein. Means the DNA contain information for the synthesis of protein, and this process is achieved by this intermediate that is RNA. So RNA के through message को carry किया जाता है to form protein. So this unidirectional flow of information from DNA to protein through an intermediate that is RNA is known as central dogma of biology. Sometimes, in some cases, some viruses contain genetic material as RNA. So this RNA can be converted back into DNA by the process which is known as reverse transcription, and the enzyme used for this is reverse transcriptase. Next important concept is reverse transcription. This was discovered by Baltimore and Tamin in 1970, and this process is also known as Taminism. In this case, it involves formation of DNA from RNA by the process of reverse transcription, and the enzyme used for this is reverse transcriptase, and this process is known as reverse transcription. So recently in COVID testing we have heard the term that is RT-PCR. So this RT-PCR uses this basic principle, that is formation of DNA from RNA by this process that is reverse transcription, and the enzyme used for this is reverse transcriptase. And this was all about for our today's discussion about DNA replication. Hope you will understand this concept of DNA replication. If you have any questions, queries, and any suggestions, you can give it in the comment section. Thanks for watching. Have a great day.